So in introducing our next speaker, it is tempting to tell you that he was once a New York City cabbie, once held the Guinness World Record for joggling, which is juggling while running, for those who've never done it before, that he co-authored and published a book of short stories in Spanish, that he makes his clothes, or that he is an ordained minister and officiated my wedding. Or I could describe the contributions he's made to OHBM, which challenge us to grapple with the post-Renaissance schism between art and science, promote openness and collaboration, and encourage us to be more social. While it is tempting, it is probably too early to compare him with Dylan, Joplin, Hemingway, Erdash, Cajal, or Broadman. Although, just like Sinatra, he has unapologetically done it his way. But I will not talk about those things because they distract from the considerable contributions he has made to our field. From his early work illustrating functional connectivity variations across the anterior cingulate cortex, a visualization I still hang on my wall and force my students to memorize, parcellating the precuneus in humans and monkeys, developing methods to improve mapping the ischemic penumbra, yeah, you may not know about that one, to his current work, understanding the functional organization of the brain, he has made many significant contributions to our field. Importantly, he is also a historian. Working with social scientists, he has recorded the neuroscience zeitgeist, even producing a wonderful film about his favorite phenomenon, resting state functional connectivity. If you haven't seen it, you really should. It's amazing. Daniel brings a unique combination of scientific rigor and collaborative spirit to our field, and we are all better for that. Without further ado, please join me in welcoming my friend, this year's winner of the OHBM Young Investigator Award, Daniel Margulies.
Thank you, Cameron, for such a warm introduction and for loosening me up. It is such a thrill to be able to present my research here today, and I want to thank the program committee for the opportunity to do so. And thank you for coming. As I mentioned, this is a line of research, which I'm going to focus on presenting here, that's been very close to my heart over the past several years. And it's what got me off of writing the whole Spanish short stories and the whatever mishigas that was going on otherwise. Focus on trying to understand what is this web of connections? How do we make sense of the patterns of connectivity, the patterns of activation, the different functions that are implicated, and as they're distributed across the cerebral cortex. And to begin, I like to go back to a historical example that I think is quite pertinent to where we are as a field. And if you'll bear with me for this metaphor, I think it's relevant. And it deals, again, with these principles of spatial organization. How are different features spatially distributed? If you look back about 100 years, multiple fields were asking independent questions. Each of these subfields was dealing with it in their own territory and on their own terms. In geology, there were very similar mineral formations that were observed across the continents, and nobody could explain why they were distributed the way that they were distributed. On the other end of the spectrum, in biology, there were very old fossil formations that were found that demonstrated the existence of similar organisms at very far spatial distances. And there was simply no theory, no model, to be able to account for these very phenomena. Everybody thought it was unique to their field. And then along came Alfred Wegener and proposed a theory that I think we're all very familiar with today of continental drift. And this model achieved a few things. It provided an underlying model to explain these various phenomena, but it also helped these different fields to recognize they had a common underlying question, that they were connected. And even though all the aspects of it experimentally didn't turn out to be true, it helped to link and provide this common model. In many ways, we are at a similar position in the field of human brain mapping at this moment. The past several decades have brought incredible innovations in the ability to map features of the cerebral cortex. Going back over the past century, the innovations in looking at cortical microstructure, to connectivity, to give one example, as well as function, just to give an example of the distribution of semantic categories. And we are at a moment now where it becomes possible in addition to addressing the question of where are the different areas and where are the different territories, where are the boundaries across these features, we can ask a question of why do we observe this consistency of these features across the space of the cortex? And in fact, this is not a question that we're posing just now. If you go back about 50 years in literature, we have a Wegener-like character in our own field, and that's Friedrich Sinides. And Sinides was trained by the Vox. He was really working with cyto and myeloarchitectonic data. He was looking at how we can subdivide the cortex into different divisions based on these features of the cortex. But in addition to doing that, he asked an additional question that was fascinating. He asked the question of why do cortical areas arrive at their specific spatial positions? What are the overarching trends in cortical features that we can use to try and understand both the evolution and the development of the cortical sheet? And over the course of several dozen papers over around 30 years, he developed this theory, which, to summarize it in one line, can be termed the dual origin of the cerebral cortex. And I'll present that theory, just a general schematic of what he was going for with that, because I think both the theory itself is important, and again, although all the aspects of it didn't turn out to be true, it's the question that he posed that was so critical and meaningful and offers a real insight into how we can think about multimodality and consistency across space. So to give the basic idea of the dual origin of the cerebral cortex, it's the idea that over the mammalian evolution of the cortical sheet, it emerges from two spatial trends of progressive laminar differentiation, meaning that there's increasing laminar complexity in the layer structure of the cerebral cortex as you move from two origin points, one of which is the hippocampus on one side, termed the archicortex, and on the opposite end of that spectrum is the piriform cortex, or olfactory cortex, so sensory-related, on the opposite end of the spectrum, and that as the cortex evolves, as it expands, we have more complex laminar structure until these two trends arrive on the lateral wall at the point of convergence between the gray and white zones that you see in the picture before you there. Now, Sinides was an interesting character. He ended up spending a year in Boston and in the lab of Deepak Pandya, collaborating with him, who's one of the world experts in macaque-monkey neuroanatomy and connectivity. And during that year of collaboration, they had the opportunity to integrate this notion of laminar differentiation and waves of laminar differentiation. How's it going? Laminar differentiation. Oh, thank you. Sorry about that. Laminar differentiation, to integrate that with connectivity, which was Pandya's expertise. And one of the very, I think, pithy principles that arose from that collaboration is the notion that like connects to like. So areas that show similar degrees of cytoarchitectonic differentiation preferentially interconnect with one another, which demonstrates now a relationship between the spatial trend in architectonics and a spatial trend in connectivity, as demonstrated here. Marcel Messelem was another very well-known researcher to pass through that lab. And for those who don't know this review, it is one of the landmark reviews which I highly recommend to anyone at any stage in their career. It's a remarkable piece of work because it's able to so fluently integrate across multiple literatures, across scales of neuroscience, from really the molecular up to the clinical and lesion studies. But part of what is put together in this review, again, extending this theory of Sinides, is the notion of a global organization, a global hierarchy across the cerebral cortex, which can be summarized in this schematic image, which he proposes in the human based off of the track tracing work in the macaque monkey. And that is sensory segregation. So you see primary visual, V1, primary auditory, A1, and those are segregated in terms of their processing. But then they integrate along these concentric circles towards a transmodal core of this organization in hierarchical steps. And that essentially provides a global organization which we can use to understand different processing streams as they integrate towards a core network of interaction across different systems. But I think one of the challenges that we currently face is, in many ways, we've really dramatically benefited from the network-level understanding of cortical organization, to give one of the more prominent examples that we're all familiar with here. But how can we think across this notion of global networks and independent functional systems and how these integrate into these global processing streams? How can we try and find a cross-dialogue between these ways of understanding cortical organization? And that's part of what I'll be addressing today. So just to give the summary of where I'm going to be going over the next few minutes, what are the spatial principles that underlie the large-scale organization that we observe across modalities? And to get at that question, I'll give a little bit of my background. I started in resting state MRI. This is demonstrating a bit of that activity that we observe and these very slow fluctuations. And the approach that I've taken to it over the past decade or so has been to try and describe functional subdivisions within complex heterogeneous regions of the cortex based on their patterns of functional connectivity. So to use this to try and characterize with a high degree of spatial precision cortical connectivity patterns. And as Cameron kindly mentioned, anterior cingulate was our first work with Mike Millen. And differentiating areas by systematically placing seed regions within the anterior cingulate cortex. We then used the macaque-monkey track tracing literature with Michael Petrides to investigate subdivisions within posterior medial cortex. And this is just a schematic demonstrating some of the divisions we observed there. And then more recently, in some work that I'll go into in a bit of detail, because it links up with the approach that we eventually ended up taking to address the question of convergence across these modalities with the space of the cortex, looking at the ventrolateral prefrontal cortex. And finally, in work with Joachim Boetger and Esther Jacobson, developing methods for high-resolution mapping of areas based on manual delineation of areas within Broca's region. Okay, so to give a little bit of a very quick overview of the work on the ventrolateral frontal cortex, this is a schematic of areas within the macaque monkey. And here we've schematized, based on the macaque track tracing literature, patterns of connectivity within that region. One of the key features is that as you move away from the central sulcus, the distance from the central sulcus, areas are preferentially interconnected. So it's got this nice rainbow kind of pattern as you get further away. So by the time you arrive at area 45, moving anterior from the central sulcus, it connects all the way back to area PG, and in addition, there's extensive connectivity within the superior temporal sulcus in the macaque monkey that's been observed. And again, this is our gold standard for understanding connectivity, cortical connectivity in the primate brain. So what we did was to, based on that hypothesis, to take seed regions within morphological landmarks of areas 44, 45, 6, and 4 within the ventrolateral cortex in 6 individuals. We had an hour of resting state each. And across those 6 individuals, we found these beautiful patterns that corresponded really nicely to the macaque-monkey track tracing literature. And we were able to see this kind of nice color progression as we move away from the central sulcus of yellow to blue to green and to red. And in addition, we have extensive connectivity of the 45, the red region within the superior temporal sulcus, so everything converged nicely. What we didn't expect was to see that same basic color progression, that same 4 ordering of yellow to blue to green to red at several other points across the cortex. And it seemed that there was this basic topographic repetition of connectivity patterns all over. And this is just to give a couple examples of that. And it raised this question for us of how could we try and observe this ordering, the spatial ordering of connectivity that appears to be present without having to assume the specific number of networks, without having to assume the starting location of them through seed regions. And it led us on a little bit of a wild goose chase for finding methods to be able to characterize this. And I was very lucky to encounter a group of researchers and have the support of Satra Ghosh and Georg Langs, and also to have wonderful interactions with Leonardo Serliani and Kun Hak, Christian Beckman as well, that led us to a set of methods for trying to characterize the space of connectivity. And I'll show you in a very schematic way what we were doing here to try and convey the method that we took. So what you see on your right is a connectivity graph schematic. It's purely a schematic, but what it illustrates is the idea that if we represent every point along the cortical surface as a point in that graph, we can then place them spatially so that regions that are highly connected or have very similar connectivity patterns are closer to each other. Right? So more highly connected, closer to each other. And traditionally, the way to characterize different networks would be to look at different modules in that graph, to subdivide it so that areas that are more connected with one another are within the same module, and if they're less connected to other modules, they're separate. But instead, what we wanted to do here was to look at the overarching location of where these patterns are in relation to one another. So we can use a class of methods called nonlinear dimensionality reduction approaches, very similar to Laplacian eigenmaps, called diffusion map embedding, to essentially pull out the dimensions of variance in that graph, the principal dimensions that demonstrate unique connectivity patterns. And along that dimension, it represents how far those connectivity patterns are from one another, how dissimilar they are. We can pull out multiple dimensions of that, and we can then project the data into that low-dimensional embedding space. So if we do that with the Human Connectome Project data, And at this point, there were 820 individuals in that data set in the functional connectivity matrix. What we see is a very similar pattern to what Meslin proposed based on the track tracing work in the macaque monkey years ago, but hadn't been able to show in empirical data yet in humans, and we're able to find it here. And so we can already see that along gradient 2, or the dimension representing the second most variance in connectivity patterns, there's a differentiation of sensory modalities. So we've got M1, S1, A1 on one side, and V1 on the opposite, and that those integrate towards a set of regions in red, transmodal cortex. If we then expand gradient 1, or the principal gradient, along the color bar, just to see where it actually terminates, what we find is that it terminates at a series of regions. At the far extreme of that gradient are a series of regions that spatially converge with what we would describe as the default mode network in the human brain. And so it's establishing the position of the default mode network at one end of an extreme that extends between unimodal and primary areas on one side, and these transmodal regions on the opposite end of the spectrum. We can then position, for instance, the canonical networks of Thomas Yeo, Fennekrina and colleagues along that gradient, and it shows a clear segregation of unimodal regions at one end in blue and purple, followed by the attention networks, the frontal-parietal control system, and the default mode at the far end of that spectrum. Then we can schematize this. Similar to how Meslin laid out that pattern, it begins to establish a spatial relationship or a connectivity relationship between these various networks. So where we've gotten up to this point now is by providing a low-dimensional embedding of the connectivity patterns. It allows us to schematize network organization as an integration from unimodal cortex towards transmodal regions. But what I think it additionally contributes from what Meslin initially proposed is it positions the default mode network along a continuum. I think it's been one of the major challenges for our field of what is the role of this system that appears to do something so categorically different than every other network in the brain. What is its role, and how do we think of it as being along a continuum with other regions? Because we can't simply excise it and consider it something completely independent. So it establishes along this spectrum, and we can begin to consider what the functional role of it might be as well along that spectrum. And of course, the only reason Meslin didn't include it at the time was that it hadn't yet existed in the literature. So now we can begin to think of how this might relate to these additional regions and to a global organization that we observe here. In addition, I just wanted to mention, because I think we were quite excited to find this and hadn't mentioned it in the original publication, but when we did the analysis, we did include subcortical regions, and we found that the ordering observed along the principal gradient converged beautifully with the prior network publications that have been observed within the cerebellum and the striatum. And I just wanted to point out that there's some beautiful work that's already been put online and posters available that are also relating to the cerebellar and hippocampal organization that I have nothing to do with, but it's wonderful work, so I just wanted to mention it as well, to check out those posters. Convergence with cortical microstructures. So at this point, we've dealt with connectivity, but I promised you some multimodality, and I got to deliver on that. So as I mentioned during the introduction, there's this principle of like connects to like, and this is the idea that areas which show a similar degree of cytoarchitectonic differentiation preferentially interconnect with one another. It's been observed and demonstrated across multiple different species, but as you can imagine, because of some of the challenges in acquiring both microstructural data through histology as well as connectivity data in humans, it hasn't yet been demonstrated. And that's precisely the project that Julia Hutenberg, a wonderful graduate student in my group, took on. In brief, she took quantitative T1 maps as a proxy for intracortical myelin density, and intracortical myelin has been used over the past century as a way of differentiating different cortical regions based on that characteristic within the laminar structure of the cortex. It was validated as an approach to map to myelin density within the cortex in Leipzig, among other places, recently. And of course, it's been used in cortical parcellation using a slightly different measure of T1-T2 weighted ratio. What Julia did was to acquire high-resolution data across 8 subjects on the 7 Tesla scanner in Leipzig. Here are the T1 maps, which is the inverse of myelin density, and to relate that to the principal gradient as it was measured in these same individuals using high-resolution data. And we found a nice relationship between these two patterns. So there's a correspondence in this case of the principal gradient in functional connectivity and the myelin density. But there were also regions that didn't perfectly match that specific relationship. And that's been one of the ongoing questions that we've been considering. But one of the interesting phenomena that we can use to try and interpret why the difference also exists is that there's been prior publication that the regions which show a mismatch specifically have a different mix of the, let's say, the proportion of long and short distance connections within that region. And that's been previously shown in work by Zuo and Sepulcre, where specifically in the regions of mismatch within the T1 map, within the myelin density, that may account in part for why we're observing differences in the match. So work for further consideration, but I find that quite provocative and interesting. How does this organization emerge? And up until now, we have these spatial patterns. But if we think back to the continental drift theory, we would like a kind of story to help us understand why on Earth all of these patterns arrive at the specific spatial positions that they arrive at. And one of those stories, which I found very compelling and which actually motivated a lot of this research, was from a wonderful review by Buckner and Kreenan in TICS a few years ago, in which they proposed a tethering hypothesis to account for the dramatic expansion we observe in association cortex within the human brain. And in brief, for those who haven't had a chance to see the article yet, it's really good reading for the flight home. If we go back to the ancient mammalian cortex, one can account for the spatial distribution of primary areas by signaling gradients during embryogenesis, which determine the position, and then those are reinforced by thalamic inputs and hierarchical connectivity patterns between these regions. We get a nice sensory layout where everything's tightly packed. But then we've got dramatic cortical expansion by the time we arrive at the human, and we have the same molecular gradients during embryogenesis, and that still determines the spatial location of primary areas, but there's all this additional cortex whose function isn't determined by the gradients, it's not determined by the hierarchical connectivity patterns within those specific unimodal or even initial kind of cross-modal areas. And what instead determines the specialization of those regions are the cortical-cortical connectivity patterns between association cortex there. They're untethered from the gradients that determine the specialization of primary regions, and hence the tethering hypothesis. What this suggested to us when we read it was that if this is correct, one of the implications we could assume is that the spatial position of association cortex should be really profoundly related to the location of unimodal regions because of the expansion away accounting for the position of each respective category. So what we did to look at that was to take the peak regions of the, or the extreme end of the principal gradient that we observed in connectivity, which you can see here, converging with the default mode network, and to calculate the geodesic distance of every point along the cortex to those regions. So what you're about to see in the next slide, and just to clarify, is purely based on the structure of the cortex. And I just want to emphasize that point because it's weird how much they relate to each other, as you can imagine. So this is purely looking at the distance along the cortex from these peak regions. And here's the map that we get after inflating the cortex so you can really see into the cell site. What you're observing is the geodesic distance along the surface away from those regions, those circles in white there. And the gray lines, or the white lines as they appear here, are the positions that are equidistant from the adjacent transmodal peak regions. What we were struck by when we first saw this is how precisely and really exquisitely those align with the morphological landmarks of primary sensory areas, the central sulcus, the calcarine sulcus, and the transverse sulcus. And it appears that spatially, the cortex is arranged so that these regions that are involved in such different functions, these regions that are systematically quite distinct from one another, are as spatially as far as you can possibly get from each other along this complex spatial arrangement of regions in the cortex, and really precisely so. It also suggests that we can begin to think of this intrinsic connectivity, if we interpret geodesic distance as representing that, as an organizing mechanism for the localization of function. And here I refer you to a couple of really nice articles, as well as posters by Conrad Bagstiel, that try to characterize and think about the implications of geodesic distance for acting as a kind of hierarchical representation as we move away from sensory cortex. In addition, I think it's worth considering how geodesic distance, as representing intrinsic connectivity within the layer structure of the cortex, might represent one of the processing features, or where we could think about computational features within, for instance, a predictive coding framework or otherwise. Within these models, we've often considered cortical organization and the, let's say, primary building blocks as being distinct areas or modules which perform homogeneous function. But what this precise mapping and spatial relationship between the location of transmodal peak regions and unimodal cortex might represent is concentric circles of processing along the surface that aren't necessarily bounded into distinct processing areas or modules, but are continuous as you move along the cortex. And this is simply an idea and thought and something that we're further exploring. Okay, so we have this mapping now between the geometry of the cortex, the actual structure, and its connectivity, a relationship with microstructure. And if all of these things are interconnected in the way that we're thinking about, well, it should be conserved across phylogeny. It should represent an axis of cortical expansion that is conserved. And in order to begin thinking about that, we looked at one of the, say, closest primate models that we have a lot of data on, the macaque monkey. Using the COCOMAC database, which is a meta-analytic database of structural, so track tracing connectivity in the macaque monkey, we observed a similar principal gradient of cortical connectivity ranging between unimodal and transmodal regions as depicted here. And in fact, we found a similar relationship with distance from the transmodal peak regions in this case. And just wait for some very excited forthcoming work by Ting, who working with Mike Millam in New York. So we found a similar relationship in connectivity distance in the macaque monkey with humans. One would imagine at this stage that we have this convergence across different modalities, across geometry, et cetera, that there are some functional implications to this organization. We can begin to think of how does this provide the building blocks of the distribution and the spatial distribution of function. And where better to go to some of our wonderful meta-analytic databases, such as Neurosynth. We also conducted this analysis on BrainMap for BrainMap fans out there, but I'll present the data today in Neurosynth. What we did was to take 5 percentile bins of the principal gradient and to simply plug that into the topic terms available through Neurosynth. And here they're laid out in a way that you see a nice diagonal along that matrix because I used the weighted mean to order them. So on one end, as you would expect, sensory and motor functions are located at the blue end of the gradient, followed by domain general processes such as attention, inhibition, working memory, control. And finally, at the far end of the gradient are these higher order abstract functions, abstract representations, implicating memory-related processes, declarative memory, autobiographical memory, semantics, social cognition. And I think as we all know here, we have to be very cautious about interpreting the specific relevance of any individual row within this matrix. But the overarching pattern that emerges is increasing abstraction from unimodal inputs towards higher order representation and integration across these different systems, with memory systems being located at the opposite end. So one can think of this as an externally oriented system at the top, moving towards an internally oriented, memory-related set of systems at the opposite end. And part of what we've been interested in further investigating in work with Johnny Smallwood and Beth Jeffries at York, who have continued to be the most wonderful collaborators you can imagine. In ongoing work, this is by Charlotte Murphy and is currently in submission, looking at an n-back task as an example of content that's generated either from perception in the zero-back condition or from memory in the one-back condition, where the individual during a decision-making process has to recall the information from memory. Now, as you would imagine, the maps that emerge are similar to what would be observed in other studies, but what we want to know is how this projects onto the principal gradient, whether this provides a reduction of the space along a single dimension that allows us to interpret these different modalities of processing. And here's what we find. So again, if we sample now those statistical maps without thresholding, but simply sample them along the principal gradient and represent the activity in the zero and the one-back condition, so in the perceptually guided and the memory-guided decision-making, we find that during the zero-back condition, there's more activity in the blue regions, in the unimodal end, as you would expect, and during the memory-related processing, or one-back condition, it's at the opposite end of that gradient. So it provides a basic reduction of this distinction in the modalities of input during the decision-making process. And in other ongoing work, in a paper actually published this year in Neuroimage, we also look at category processing as it's involved in both memory and perceptual decision-making. Another line of work that I think is really just so elegant and exciting is by Boris Bernhard, a wonderful collaborator at MNI. And his group has just been taking this gradient stuff so far forward. It's been such a thrill to watch what's come out of the group there. So this is just one very quick example, and I urge you to go and check out the poster, 1171, where we find a difference in how we interpret is the hierarchical structure with an autism spectrum disorder, in contrasting the principal gradient with controls, as you can observe here. So it's a more constrained principal gradient. And I urge you to check out the poster for further elaboration of that work. So overall, we can begin to think of network organization and the basic spatial relationship between these systems as a kind of recruitment along this gradient that's occurring, where we have networks that are organized topographically from primary or unimodal towards these transmodal or associative regions. The distance along the cortex seems to be critical for that spatial organization. And in a really interesting way, there's this isomorphism between the structure of the cortex and the functions that these regions subserve, such that internally and externally oriented processes are maximally distant from one another. There's this exquisite relationship between the location of these systems over the cortex and the functions that they have. This is just a summary slide, which I won't go through in detail because I've already presented it, but it summarizes some of our work addressing the question of how we can characterize what we think of as a first dimension of a kind of connectivity space or an intrinsic cortical space that seems to capture something that's both neurobiologically meaningful about the cortex across these modalities, but one that also has functional implications. And if we think about this in conjunction with the second gradient, in a wonderful review by Julia Hudenberg, who I already mentioned, who is a graduate student, is now at Champollion in Portugal, we've begun to think about the problem of how this could provide an intrinsic coordinate space. And we're very much at the beginning of this agenda, but one of the first proposals that we put forward in this review is the notion of using the geodesic distance along the surface that I already mentioned as providing a first coordinate system. This is looking at the distance from the transmodal peaks. And then we could imagine that relative distance from the different sensory systems, the morphological landmarks of the sensory systems, provides a second dimension that we can use to localize regions across the cortex. And of course, this is an area of ongoing research, but it's an exciting one for us because we're beginning to think about how we could represent this on an individual level in a way that's both meaningful for multiple modalities of cortical organization and based off of the features of the cortex itself. It's not imposed from the outside, but emerges from the structure of the cortex. Okay, so up until now, I've really focused on describing just really the first gradient. The second one seems to be quite, with all respect to the second gradient, it seems to be quite obvious in terms of how we interpret it. It's differentiating these sensory modalities. And we've taken it slow for the reason that it's easy to pull out all sorts of different gradients. And that's what you're seeing in the upper right over there, is the variance explained by the different gradients. And we can pull out dozens and hundreds of these patterns, but one of the challenges becomes how do we integrate this into our vocabulary within cognitive neuroscience? How do we use this and kind of bring it in as a structure for characterizing the patterns of functional activity, the mechanisms of different cognitive states that we're interested in characterizing? And so we've tried to take it very carefully in offering an interpretive framework, an explanatory framework for each of them as we come to understand them. And so what I'm going to show you now is what the third one looks like as a kind of addendum. We really got the rendering farms going for this next video, so, buckle up. You're welcome. Yeah. Anyways, I'm going to color them now with a different, I don't know. Anyway, I'm going to color them now so that you see the actual spatial layout of that spectrum from the third gradient from the top to the bottom. And what you probably recognize on your right is the regions in yellow being a kind of multiple demand network, to use the Duncan terminology, or a kind of collective attention network, to take some other vocabularies into account in describing the distribution of these regions. So now we have these three different gradients, and we can begin to use that to try and predict task state. And this line of work, I think these findings in many ways are something that has been addressed by multiple groups, so I'm just showing it. It's ongoing work by a wonderful postdoc, Marcel Falkiewicz. And what we did was to use those first three gradients as spatial regressors in a general linear model and to characterize data from the Human Connectome project in the NBAC task. And what we can do then is for each individual is to project each volume into a space described by each of the first three gradients. So it's very similar to kind of a dual regression approach, if you're familiar with that. We can then average within each of the task states. So for each individual, we can now project each of their task states into a three-dimensional space. And that's what this looks like. So what you can already see in this three-dimensional space is if you just take two of the dimensions, for instance, gradient 1 and 3, you can visually very clearly segregate the different conditions. And of course, we were able to have high prediction accuracy, as you would imagine. Each of these dots represents a single individual and a single condition. But taking that a step further, we also found that it was the location within this gradient space, especially along gradient 3, that correlated with the task performance in different directions in the 2 and the 0 back, but it related in both. And so we can begin to interpret the role of gradient 3 as representing a kind of functional dimension between working memory and the input of content at the opposite end of that dimension. This is ongoing work, but just I wanted to present it as a way of where we're going forward, which is a model-based approach for functional organization. How can we think across these modalities? How can we understand the basis of how these patterns emerge and offer a converging framework across these different approaches to measuring brain organization, the features of the cortex? In addition, it provides a kind of spatial model or a way of thinking about these axes, these conserved axes of expansion across different species within mammalian phylogeny. And finally, I think really critically is how do we think about a neurobiologically meaningful space for representing functional specialization? How does the space of the cortex and the distribution of different regions offer us insight into the possibilities and the constraints that we have of cognition and how we experience the world? And so with that question, I'd like to thank so many people and institutions. This is my chance to really thank everybody, and a lot of people do deserve thanks. Of course, Marcel and Yulia, who did a lot of the work, Chris Gorgolewski, who's just an amazing, amazing person, Alex Goulas, who also has imparted a wonderful love for neuroanatomy, Smedar and Shema and Franz and Manusos, Estrid, Johannes, Sabina, Mark, Wajay, Philip, and Natasha, who held me together. And also former members of the group, Yeting and Xiangyu, Alex, Melissa, Judy, and Joachim. And then my collaborators, who are some of the most wonderful, encouraging, supportive people you could possibly imagine and want both as friends and colleagues. Johnny and Beth, you guys know, we couldn't have done any of this without you, and it's been such an absolute pleasure. I look forward to many years of shared collaboration. Satra and Georg were really there to provide incredible methodological and moral support. Simon, Michael Petrides, Boris, such beautiful work. Gleb, Pierre-Louis Bazin, who did a lot of the collaboration with Julia Hutenberg on the high-resolution 7T data. Cameron, yeah, you know. And Javier, thank you for everything. In addition, I want to point out a few posters that are related to the gradients, and really most of them I had absolutely nothing to do with, but I just thought there's some wonderful work that I would recommend checking out if you want to know more. And Kun Hak, who I think I saw over there, hi, did us all a wonderful service in the community by releasing the connectivity gradient, or Congrads code, recently. And so it's up on GitHub. And there's also going to be a presentation tomorrow at 12:30 by Christian Beckman. And this is really one of the origins for the methodology that I presented. We use a slightly different method here, but it's really one of the more powerful toolboxes and approaches for addressing these kinds of questions. So if you're interested in finding out more about the methodology and how you can do this at home safely, go and check that out tomorrow. Finally, just shameless plug for Shubigi Rao's art exhibit, which is just extraordinary this year, and right over to the left as you exit. And I highly encourage everyone to check it out because it's absolutely beautiful work and asks some interesting questions about language and the brain. And in addition, you can find our software and data over there. The maps are up on Neurovault. Thank you to the Human Connectome Project and Prime. And thank you for listening and putting up with this. I really appreciate it.